from University of California, San Francisco. First, I'm going to thank the Cancer International webinar for their kind invitation. Today, I'm going to talk about the therapy-induced evolution of non-cancer revealed by single-cell RNA-seq. As we know that the cancer is currently viewed as a disease of involving genomic instability and abnormal epigenetic modification. The divergent complexity of tumor genomic and functional heterogeneity contribute to the tumor initiation, progression, metastasis, and the response to the drug treatment. Unfortunately, the subpopulation of cancer develop resistance and a tumor relapse. This is the common challenge in the cancer research. Since we focus on non-cancer, just to give you a brief background, non-cancer is the leading cause of cancer-related deaths in the United States and worldwide. There are two different histopathological types, small cell non-cancer and non-small cell non-cancer. Depending on the stage of cancer, various treatments are available. For advanced non-small cell non-cancer, so targeted therapy, the immune checkpoint inhibitor treatment are the main choice. For targeted therapy, particularly EGFR mutant non-cancer, the patient initial response to treatment followed by relapse after 9 to 12 months. For immunotherapy, only a small proportion of patients benefit from that. So the intrinsic or induced drug resistance is a major hurdle for successful treatment. There are several models that have been proposed to account for the emergence of drug-tolerant persistent cells in clinical we call minimum residual disease. So here is an elegant review paper illustrated the three different models. The Darwinian selection model posits that the rare drug-tolerant cells exist in tumors before anti-cancer therapy. So those cells were selected and survived, developed the minimum residual disease. So recent studies also suggest the non-marking induction hypothesis. So in this model, suggests that the drug-tolerant cells arise from heterogeneous cancer cell population through the epigenetic modification upon treatment. So those cells stochastically developed to minimum residual disease or drug resistant. So the third model is called dynamic fluctuation. So with the notion that the expression of resistant markers fluctuated in cancer cells, at the treatment initiation, the rare cells, so the rare cells expressing high level of various resistant genes will survive and reprogrammed to stably express the resistant signatures. So those cells will develop residual disease or persistent. So regardless of different models, the drug-tolerant persistent cells share common features, such as slow proliferation, the highly flexible energy consumption, adaptation to their microenvironment, and the phenotypic plasticity. So recent study also suggested the rare high proliferation cells can develop drug resistance. The complexity of tumor microenvironment also suggests the interplay between the cancer cells and the host immune cells. And the crosstalk between the cancer cell and the many molecules derived from the cancer-associated fibroblasts, macrophages, endothelial cells also play an important role in the development of drug resistance. So to understand the underlying molecular mechanism of treatment-induced resistance, we employ the single-cell RNA-seq technology to serial biopsies from advanced non-cancer specimens. So we obtained 49 samples from 30 patients. Those samples were grouped into treatment-naive residual disease or progressive disease. So the common oncogenic drive gene mutations were detected at the diagnostic stage, such as EGFR mutation, ALK, BRAF, and KRAS. So those samples were dissociated and sorted based on CD45 labeling into immunocell and non-immunocell. So those sorted cells subject to SMART-SIG2 platform for single-cell RNA-seq. So at the end, we obtained over 20,000 single cell, including epithelial, immune, and stromal cells. So this is an overview of the data analysis. We first focus on the epithelial cells and categorized into cancer and non-cancer cells based on copy number change. And then performed the mutation analysis and the transcriptional analysis between the treatment groups. We further analyzed the immunocell within the tumor microenvironment. So all those findings were independently validated using independent data cohort, as well as experimentally validation. Given the association between the cancer and larger scale chromosomal alterations, we utilized the copy number variation from RNA expression to classify epithelial cell either as cancer cell or non-cancer cell. So compared with the fibroblasts or endothelial cells, so the cancer cell displayed a larger change from relative expression intensity across the genome. As shown here, the copy number gain in red color and then copy number loss in blue color. All the cancer cells were classified, resulting in 25 unique clusters. So when we calculated the number of cells of the highest contributing individual patient over the total number of cells, so as shown in the panel C, the majority of cancer cell clusters are the patient-specific and having the high patient occupancy score. In contrast, the non-cancer cells show the blue lines, which are pretty much evenly distributed across the patient has low patient occupancy. That's indicated the extreme heterogeneity of cancer cell from different patients. So mutation analysis. So many genetic alterations can coexist with the primary targetable oncogenic driver alterations such as oncogenic EGFR, ALK, KRAS. And those oncogenic driver genes may help promote tumor progression and limit therapeutic response. For this reason, we acquired a single-cell RNA-seq transcript from each cancer cells as shown in each row to identify somatic mutation as many as possible. So in the panel B show critical driver mutations detected at the diagnosis and also able to confirm at the single cell level. In addition, many cancer-related gene mutation also detected at the single cell level as shown in panel C. So just to give you one example, the patient LTS47. So initially, this tumor have ALK fusion with EML4. And those fusions were recaptured at the single cell level. In addition, the KRAS mutations also detected at the single cell level. and many those cancer-related genes mutation at a single cell level. So this suggested the intratumor genomic heterogeneity may be underestimated when using the DNA bulk sampling. So at the single cell levels, the occult genetic alteration or subclonal mutations can be revealed. So the impact of those mutation profile is profound. When we perform the Kaplan-Meier survival analysis, we show that the patient with more concurrent mutations tended to have poor survival than the patient having less concurrent mutations. So here we focus on the cancer cell with a transcriptional profile. We performed the different group comparisons between the treatment-naive residual disease and the progression disease. We're able to detect alveolar signatures highly expressed in residual disease, indicated the alveolar type 1 and the alveolar type 2 associated genes high expressed. Meanwhile, proliferation marker genes expression decrease indicate persistent cancer cell state at the residual disease. So when we compare the treatment-naive between the progressive disease, the chirulinin signature gene highly expressed in progressive disease, as indicated IDO1, KYNU, and KOPRT gene high expressed indicate immunosuppressive behavior. And when compared the RD versus PD, the plasminogen activation signature high expressing in PD group, indicating that the tumor cell gained the invasive capacity and the cell-cell communication and the immunomodulation functions. So here we illustrated the longitudinal sample analysis with the single cell RNA-seq. So as you know that the obtaining the consecutive critical tumors biopsy from individual advanced stage lung cancer patients before and during treatment is very challenging. We obtained series samples from the same primary tumor site from 3 different time points: before treatment, during treatment, and when the tumor regrowing. So in all 3 biopsies, we identify EGF exon 19 deletion drive mutation in cancer cells, as shown in figure D, and additional cancer-associated genes mutation detected in different samples. So more importantly, we observed that the cell type switching from initial the non-adenocarcinoma, as panel C showed the HE staining, and then switched to squamous cell carcinoma. So this is carefully examined by experienced pathologists. Also, the squamous cell differentiation genes are high expressed at the single cell level. So the cancer cell type switching is one of the drug resistant mechanisms in EGFR mutant lung cancers. So we next addressed the evolution of the tumor microenvironment during targeted treatment. As shown here in left T-SNE plot, the immune cells were clustered and annotated. So the immune cell type clusters show low patient occupancy. And when comparing the immune cell composition across all 3 time points, as shown the right panel, so using the fraction immune cell abundance, we observed a significant dissimilar pattern in residual disease relative to TN and progressive disease. So as I highlight here, the macrophage monocyte, the abundance is lower in residual disease compared to TN and the PD. In contrast, the T cell abundance is high in residual disease relative to TN and PD. So then we focused on the macrophage subpopulation analysis. So the macrophage from the non-tumor biopsies were further divided into 5 distinct subclusters based on differential gene expression, as shown here on the middle panel. And each is the subclass across 3 treatment points are different, particularly labeled, which is M2 subclass enriched in PD group, MF3 class enriched in RT, MF4 subclass enriched in TN. So panels B show individual gene marker differentially expressed across the subtype of macrophages. Similarly, the T cell, the NK cell were divided into 5 different subcellular populations, as shown in T-SNE plot. Although the T cell infiltration remained limited in progressive disease, there were relative enrichment of the T regulated cells and the exhausted or dysfunction the T cells enriched in progressive disease. The panels, the figure D showed the individual marker gene differential expressed in different subclasses of T cells. So this suggested that the immunomicroenvironment dynamic changes from the treatment naive, the immunosuppressive environment, and the change to the residual disease when tumor responds to treatment and become immunostimulatory. And based on the macrophage infiltration decrease and the T cell infiltration increase. When the tumor become progressively and the change to immunosuppressive environment again, and illustrated here the macrophage increase and the T cell decrease. So we still try to understand the detailed mechanisms, how they affect the treatment. So many data I don't have time to present. So anyone interested can read the paper we published in Cell years ago. So in summary, our project is able to demonstrate that the single cell RNA-seq is feasible in metastatic human non-small cell lung cancers and reveals a rich tumor ecosystem. So individual tumors and the cancer cell exhibit substantial molecular diversity. So cancer and the tumor microenvironment cells exhibit marked therapy-induced plasticity. So single cell RNA-seq metastatic non-small cell lung cancer unveils the new opportunities to improve clinical outcome. So finally, I would like to thank many people who contribute to this project, including the medical oncologist, the physician scientists, the experimentalists, and bioinformaticians, and also, importantly, our patients. So thanks also go to the funding agents for their financial support. So thank you, audience, for your listening. I'm happy to answer any questions you may have.